Hello, everyone, and happy Wednesday. Thank you so much for joining us for FAU's Harbor Branch Behind the Science, a Wednesday web chat. I'm Madeline Arancibia from the Outreach and Engagement Department, and I'm going to be your host today, as per usual. And today, our special guest is Dr. Josh Voss. Josh has been at FAU's Harbor Branch since 2007, and he acts in two capacities here. He's an associate research professor, and he also is the executive director for the NOAA Cooperative Institute of Ocean Exploration, Research, and Technology. He has a bunch of different research interests. A lot of them, of course, align with corals, which we'll be talking about today, focusing on coral health and ecology, but also looking at a bigger picture of marine conservation and management as well. He studies corals that live in shallow waters and corals that live in deep waters. And he uses a lot of different techniques, including molecular ecology, to really get a better understanding of these corals. So we're gonna get him joined in just a second, but throughout the chat, if you guys have any questions for Josh about his coral research or that Cooperative Institute, definitely go ahead and ask them. If you're on Zoom, use that Q&A button in the bottom center of the screen. If you're on Facebook, ask your questions in the comments and we can answer those as well. So we will get Josh joined in and just sort of jump right in here. So thanks so much for joining us, Josh.
Good afternoon, Madeline, and hi everybody out there. Thank you for taking time out of your Wednesday to chat with us a little bit.
So in your intro, I said that you do a lot of different things. You've got your hands in a lot of different pots here, and they seem to mostly sort of be revolving around corals. So what really got you interested in them? What was your inspiration as a young child or a young scientist?
I've always been a fairly science-minded kid. And as a scientist, you get to remain kind of feeling like a kid, discovering things all the time, which is fantastic. I grew up in Florida, and I was a bit of a surf bum in middle school and high school and was around the water quite a bit. And then particularly I had a science teacher, Joe Carpenito at St. Margaret Mary in Orlando, where I grew up. And he was just an incredibly inspiring individual. He challenged us in really great ways, whether it was forcing us to touch the dissected materials of things that most folks found disgusting, or throwing us all in a bunch of buses and going down to the Keys, which we did when we were in 8th grade. And that first trip to the Keys, where we were totally focused on kind of learning goals and marine science that whole time, was kind of illuminating for me in a lot of ways. And we were only there for 4 days, but it really stuck with me. And then as I started to think about kind of longer term, I think his influence as a teacher also resonated with me a bit. So I started considering kind of teaching as a goal overall. So kind of that background of both being connected to the water through recreation and opportunities to get out in nature with someone else who was passionate about it, I think were the things that really piqued my interest about science. And then I particularly remember when I was about like maybe 16 or 17 years old, and was kind of in this crossroads between, should I pursue medical science and potentially become some kind of medical professional or doctor, or natural science? And I always knew kind of from that point on that I wanted to oscillate more towards ecology and marine science. And then when I found out that you could get paid to go to graduate school in the natural sciences, I knew, like, all right, that's definitely what I want to do.
Yeah. Are you still a surfer?
Occasionally, much more of a stand-up paddler now than a drop-in surfer.
Okay. Wow. Still, I'm sure you could pull out that skill if you really needed to one day.
Of course.
And then were you scuba certified at 16 when you were trying to make that decision, or did that come later on?
I was not. So that didn't come until I went to undergrad. So when I was looking for undergraduate institutions, I was really kind of enthralled by the idea of the North Carolina liberal arts education. And so I was looking at schools that fit that mold and ultimately decided to go to Elon University, both because it had a really strong Division I soccer program and because it gave me the flexibility to pursue both biology and biological sciences and philosophy, which was really attractive to me. And then in sophomore year there, I got scuba certified through the local shop that was there at Elon.
Okay, so you're a soccer player too then. I'm learning all sorts of tidbits here. Very active. All those things that require a lot of cardio, which is my least favorite thing. So from Elon and doing that program, I guess you got your toes wet, literally, with scuba diving, and then wanted to get paid for grad school. Where did your journey take you from there?
So Elon had really great opportunities, but it was in the middle of North Carolina and not close to the ocean at all. So each summer I was really trying to seek out particular opportunities that would allow me to kind of take this amazing base I was getting at Elon and pepper in the bits of marine science that I needed to kind of be more competitive and to pursue my career goals. And so I spent a summer at Duke Marine Lab, both as a research technician and a student, and really enjoyed that. And then I applied for an internship in the Bahamas at Lee Stocking Island. And once I got that internship, that kind of completely changed my career. So I did my undergraduate research there, where we were required at Elon to do an undergraduate research thesis in the program that I was in. And so that's where I first started to focus on coral health. And then from there, as kind of undergrad started to come to a close and I started thinking about graduate school, I was really focused on places where I could kind of be at this interface of marine ecology and pathology, and coral disease was a really kind of interesting area to work in. And so I was lucky enough to work with one of the most well-known disease microbiologists, Lori Richardson, down in Miami at FIU. And so I went directly, I remember specifically like the day after, maybe 2 days after graduation, packing up everything and driving and starting graduate school directly, like 2 days after I graduated from undergrad. I would not recommend that to anyone. Take some time off and relax a little bit. But it worked out quite well for me. And because I knew that I wanted to pursue marine science research and eventually become a professor, I elected to forego doing a master's and just went straight into the Ph.D. program there. Which again is not necessarily always the best choice for everyone, but it worked out pretty well for me. And then I enjoyed Miami in my 20s. It was also a really important hub. I lived 15 minutes away from the Miami airport, which meant that kind of in the span of half a day, I could get anywhere in the Caribbean that I needed to during disease outbreaks or disease events that were going on. But I started to focus more and more on trying to get back to those locations I've been to during my internship in undergrad at Lee Stocking. And so we worked on a number of proposals to NOAA, and I was funded over 4 years to basically go out there every summer to do companion research to what I was doing in the Keys, also in the Bahamas, trying to focus on what was causing these increases in coral diseases, how are corals being affected, and what kind of environmental drivers were resulting in this catastrophic kind of event that was going on.
That sounds like a lot of school back to back with no breaks. Because once you start grad school, whether it's for a master's or a PhD, you don't have summer vacations anymore. It's not like undergrad. So you just went straight with no break. Wow. I hope that you took a nice vacation once you were done with your PhD.
Nope. Same thing. Drove directly from there to Eckerd. and started a professorship at Eckerd.
Oh my gosh. Well, hopefully you get to take vacations now. And probably some of your field work is pretty fun, I'm hoping.
It is. It's fantastic. I mean, you can see where I am right now in North Carolina, though, that because I get a chance to do really awesome field work in often tropical places, when I get a chance to get away, I typically favor the mountains and the green over the blue and the beach, just as a foil to what I do on a more regular basis.
Well, beaches work. Mountains can be fun. So, okay, your PhD was in coral research. You spent a lot of time traveling. How were your field seasons? I mean, you're saying if a disease popped up, you just jump on a plane and go. Was that like?
So sometimes we would do that for different outbreaks that were going on. So that often happened kind of early in the summer or late, September into October. The way the program was structured at FIU, it lended itself really well to taking an entire summer to do research remotely. So for 4 years of my PhD, all summer long, 10 weeks each year, I went to Lee Stocking Island. And in addition to the NOAA Coral Program grant that we'd gotten, I basically worked out a deal to work part-time as an intern there to offset housing costs. And so I would put in 20 hours a week helping to fill scuba tanks or fuel boats, and then have the rest of the time to focus on my graduate research.
Cool.
It was a very beautiful place. Anytime you can go to your research kind of, I don't know, remote from the rest of your daily job and focus on it, be it a research expedition or a research cruise or something like that, that kind of standalone time to focus on it while you're there, I think really is helpful. And in my case, being at Perry Institute and Lee Stocking resulted in an additional benefit because that's where I met my wife. So we were both interns out there at the same time. And we met in 2003 in the Bahamas. And I think a lot of people thought it was just going to be a summer thing, and it has never stopped. So that's Ashley and my two beautiful daughters right there.
What a beautiful family.
I agree, but I'm quite biased.
I mean, it looks like a stock photo that you would find like in a picture frame at Kohl's or something. I think you guys have some potential as models. So you did your PhD, you met your beautiful wife, and then you moved on to being a professor. How did you ultimately end up here at Harbor Branch?
So after a year at Eckerd College, which is over on the opposite coast of Florida, an opportunity came up that I actually thought I had missed out on. So when I was still in my PhD in around 2005, a posting was listed for a new coral researcher to come to Harbor Branch and help start this new Robertson Coral Reef program. But I wasn't done with my PhD yet. Unfortunately, right around the time of that posting was also when two massive hurricanes came through Fort Pierce and did some significant damage at Harbor Branch in particular. And so the decision was made to postpone recruitment of those positions. And by the time they were ready to recruit them again, I was at Eckerd, and I liked my position and I liked being there, but it was primarily undergraduate teaching focused. And I was attracted to Harbor Branch for two reasons. Number one, this impetus to start and grow a new program. And there had been a large donation from the Robertson family to help start a new program. And then also at that time, Harbor Branch was becoming part of FAU. And because teaching was something that I was really passionate about and I wanted to be, wanted the opportunity to mentor graduate students, the fact that we were becoming part of Harbor Branch was really attractive to me. And so for those two reasons, I left my position at Eckerd and came back across the state to Fort Pierce and to Harbor Branch. initially we thought it was going to be for maybe 3 or 4 years, and that was almost 13 years ago now.
I'm glad that we kept you around. Hopefully you stay around for a while more too, especially with all of your responsibilities with the NOAA Cooperative Institute and all of your really fantastic research, which we're probably going to jump into in just a few minutes. You've done some impressive things with coral disease.
Yeah.
But
Harbor Branch is a great place to be. I think we have a really good community of researchers and people that are passionate about the work that we do. I also think that Fort Pierce is totally a diamond in the rough that not a lot of people know about. So maybe I should be keeping my mouth shut. Hopefully our audience isn't too big. But it's a beautiful place with a good community. Now we have kind of family roots in the area as we've recruited people down, including my parents have now moved down closer to us as well. So I think Harbor Branch in particular and Fort Pierce in general are fantastic places.
All right, you're going to be a lifer here. You heard it here first.
We'll see.
So a quick reminder for our audience, if you guys have any questions for Josh, like I said, we're going to jump into some of his research experiences now. Just use that Q&A button or comment your questions as well. Jumping into some of those experiences that you've gotten to have, do you have maybe a coolest place that you've gotten to travel for research that you can think of?
Sure, I've been quite lucky to travel pretty broadly throughout the Caribbean. And one place that had always fascinated me, but had always been more difficult to get to, was Cuba. And we worked for a number of years trying to set up a really effective research partnership with the University of Havana and some of the other agencies in Cuba. And that finally came to fruition in 2017. So as part of our NOAA Cooperative Institute and this great collaboration we built with people like Patricia Gonzalez down at University of Havana, we were able to do a complete circumnavigation of Cuba, where we were exploring these mesophotic or middle light reefs that go from, just the bottom of standard scuba depths at about 100 feet down to about 400 feet in some places. So we wrapped the entire Cuban archipelago over about a month. We conducted research operations at 43 different locations, collecting thousands of samples for biodiversity and connectivity studies. And then one of the things that was the most fascinating and rewarding part of that trip was that our research crew was half and half Cuban scientists and US scientists. So we were all on the boat together, working together, leaning on one another's expertise and knowledge in ways that were really complementary and encouraging, and all continually just kind of dropping our jaws at what we were seeing. Some locations that had over 80% coral cover, some locations with huge schools of different kinds of snappers spawning together. It was just a breathtaking, beautiful, and fascinating place with a great research team. So I think it's one of the ones that, when I look back on my career much later than now, I think it's one of the ones that'll stick out without a doubt.
Everything that I've heard about the reefs in Cuba is that they are like these beautiful, pristine, almost untouched reefs, just because of the restrictions that have previously been in place for people to go and travel there recreationally. Were the Cubans that were on that trip with you more familiar with the reefs because they had gotten to experience them more often, or was this kind of new for them too?
So in terms of the shallow reefs, most of them, all of them that were collaborating with us had an opportunity to do work on shallow reefs, but few of them had an opportunity to do a lot of deeper research. So we were bringing along an ROV, or remotely operated vehicle, that gave access to deeper locations than a lot of the research that had been done previously. So I thought that was really cool. And I think too, the fact that Cuba's reefs are in such good shape is not just from a lack of access. They've been pretty aggressive about their marine management and policy. So the estimate is that somewhere between 20 and 25% of the entire Cuban coastline is protected in some way. There's also been some fairly strong restrictions against fertilizer use, which might reduce eutrophication impacts. So I think it's both a remoteness thing and an aggressive and very proactive management strategy to conserve the resources that they have.
I didn't realize that they had done all of that great management work, but that definitely is good to know, and good to know that those things have the potential to work. We can use that as sort of a goal and a guide for what we can do.
So there's actually been a work on a direct partnership for that. So they're part of our project, and part of the emphasis for our project was the sister sanctuaries agreement, where 2 sanctuaries in the United States and 2 marine sanctuaries in Cuba partnered with one another to do exactly what you just mentioned, kind of share best practices, think about how we can manage things more effectively. And I think we all kind of recognize that ocean currents, animals, plants, microbes, they don't respect whatever international boundaries or state boundaries or any other boundaries we want to put up. So when it comes to kind of effective regional management, having good international partnerships is key.
Cool. Definitely good to have that knowledge, have those collaborations, and keep working towards that. And like you said, working with our international partners. So the next sort of thing I'm interested in is another cool thing, maybe coolest animal that you've seen on your adventures.
There are lots. And this is one that I knew you were going to ask. So here's a picture of what I think was probably our best single day animal interaction that I've ever had. And this is about a 44-foot long whale shark that we encountered 100 miles off the coast of Louisiana in the middle of the Gulf of Mexico. So for scale, Mike McAllister there, who's a research technician and lab coordinator that works with Matt Ajimian, he's probably like 6'6", maybe even taller than that. And he was just completely dwarfed by this whale shark. We had finished a long transit during a research expedition, and we were hoping to just kind of tie up to a buoy that we knew in a location and rest up for the night. And anytime you're on a ship, it's easier to get sleep and to do work when the ship is tied up and moored, not running into the waves. And as we pulled up towards this mooring, we immediately saw the pattern of the whale shark in the water. So I would say half of us were in the water with our snorkel gear within 30 seconds, give or take. And you know, there's pretty important considerations for any time you're interacting with marine megafauna in terms of not wanting to impede their movement or restrict their behavior in any way. So you kind of got to stay back and, you know, so something like what Mike is using there, a GoPro on the end of a long handle, allows you to get some closer photos without getting too close yourself. The fantastic part was that we were on that mooring and we saw the whale shark kind of go off into the distance after maybe 2 or 3 minutes. And we thought, well, that was cool, but that was it. It did repeated loops down completely out of sight and all the way back up to us for almost 2.5 hours, until the point that honestly we started, it was the most spoiled moment I can think of, because we got to the point where we were bored after looking at a whale shark for 2.5 hours. But it was beautiful, majestic, kind of reminded you that our oceans operate on these immense scales that are so much bigger than we are. And a really fun experience overall. And that region of the world is just fascinating. The Gulf of Mexico is unique in terms of its topography and bottom type. Because it's semi-enclosed, it stays nice and warm and can support all kinds of biodiversity and productivity. And it's somewhat unique in that it's because the continental shelf extends so far out, most of it that is interesting habitat is all within US waters, which is also really cool.
My number one on my bucket list is to go down to Mexico during the time of year where there's the whale shark migration. Yep. And the is it manta ray or eagle ray migration that like coincide? And
I think they're around the same time. I think they come in. So what they mostly come in to do down there is to feed on spawning of other marine inverts. So things like corals and sponges are releasing all of their eggs that have lots of fat in them that are quite tasty, and all kinds of small plankton come in to also eat those spawn that are happening, and then the whale sharks and the rays come in to swoop up them as well.
I want to be there in the water with them.
I agree.
That's like number one on my bucket list.
I've not been there either. I would love to.
So that area, the Gulf of Mexico, sort of is, like you said, it's a really special place. And I know you've done a lot of work in that area too. Is there any project that really stands out that you've worked on in that area?
So I think at least for my team's research, the best part about our NOAA Cooperative Institute has been the ability to partner directly with sanctuaries and other aspects of NOAA to do really cool ocean exploration. So our Cooperative Institute is directly aligned with the NOAA Office of Ocean Exploration, or OER. So Ocean Exploration and Research, or sometimes just called NOAA Research. But we also recognize that sanctuaries have this incredible burden and need to understand the resources that they have so they can manage them effectively for all kinds of uses and needs. So in the Northwest Gulf, kind of along that continental margin, there are 3 banks that currently comprise the Flower Garden Banks National Marine Sanctuary. And for the past 10 years now, we've been able to do a lot of work in collaboration with the sanctuary staff there, including the superintendent, GP Schmal, and the research coordinator, Emma Hickerson. And a lot of that work has been focused on trying to understand what the biodiversity is in that region, particularly on deeper and middle light reefs, and then also to understand connectivity. So here's a map. You can see the 3 banks in yellow are the banks that are currently protected as part of the sanctuary boundaries. But there's all of these other banks that are spread across the Northwest Gulf as well. And over the past 10 years, we've been to most of them and done lots of assessments of biodiversity and then collections to look at molecular fingerprints to try to trace connectivity among these banks, to try to figure out which one of them may be providing larvae or fish resources that travel to other banks downstream or upstream, to come up with a management plan that can support fisheries long-term in this region, and support biodiversity for all kinds of other reasons as well, including ecosystem services, stability, etc. So the sanctuary has been really focused on trying to help preserve some of these habitats. So we've targeted a number of our research cruises to go to banks and build both video and data to support designating these as special locations that are worth protecting. And there's been great progress by the sanctuary staff and their advisory committee on that. And so now they've revised a preferred alternative, or actually kind of in that final process of starting to make a rule to expand the sanctuary to include all of these initial banks that are shown here in white. And I think it's quite likely that this is going to move forward in the not too distant future. And it's going to increase the amount of protected coral reef area in the Northwest Gulf by almost 3 times what's currently protected by the Flower Garden Banks National Marine Sanctuary. So it's habitats that are just fascinating. They support things like that whale shark that we just saw, as well as really diverse grouper and snapper fisheries that are important for the region. And so the fact that there's going to be some protections put in place for those, I think is a really important thing, because these are our natural resources. This is a shared resource within U.S. waters that we all have access to. There are other interests in the region as well, shipping, oil, and gas, and balancing those uses in a way that's sustainable and effective long-term, I think is really important.
Well, and it echoes back to what you were saying earlier, that with connectivity, it's not just connectivity between these banks, it's also connectivity with, I'm sure there's water flow that takes stuff down to the Keys and beyond. And so protecting those areas better is going to help the entire coral population, right?
It should. So we've done some assessments of how some coral species in the Northwest Gulf are related to Belize and Cuba and offshore in Florida as well. And there seems to be fairly strong connectivity between the Mesoamerican reef down in Belize, at least between the deeper corals there, and areas up in the Northwest Gulf and across down into the Florida Keys and maybe Cuba as well. There are areas that are also isolated. So for one of the coral species, there's some areas like the shallow parts of Belize that seem relatively isolated and not well connected, and other areas like Pulley Ridge off the Southwest Florida shelf that seem pretty isolated. So, both of those are useful pieces of information. You might make the argument that, well, if they're well connected, those are the ones we need to protect because they're providing larvae downstream that are important for reef development across the whole region. You may also make the argument that if those isolated reefs are providing a really important economic resource, say a really important economic fishery, that they're vulnerable because they're not getting as much larval recruitment from other places, and maybe they need even more protection.
Mm-hmm. Or just protect it all. Very
unlimited resources, maybe, but that's not quite the way it functions.
Of course not. So I have one more big question for Josh, and then we're going to get to the questions from you guys. So again, another reminder to send your questions in if you have them. But I think the last big thing that I want to touch on before we get to those public questions is if there's been a research project that you've worked on that has really surprised you.
So I'll say that after cutting my teeth on coral disease research in grad school, it was really fascinating, but it was also somewhat depressing at times because we were watching lots of corals die, and we didn't have much of a way to stop it effectively. Recently, starting in about 2014, a new coral disease outbreak started in Florida. And it's called Stony coral tissue loss disease. And it seems to be one of the worst disease outbreaks that we've seen. It affects lots of different coral species. It kills corals relatively rapidly. It's geographically spread now through most, but not all of the Florida Reef Tract from Martin County all the way down to Key West. And it's spread throughout the Caribbean as well. So it's this huge growing problem that teams of hundreds have kind of rallied together on multiple different organizations to try to understand the disease and take actions to potentially stem the tide and reverse the declines that we're seeing. So some colleagues of mine, Karen Neely and Brian Walker, have been toying with the idea of well, what can we do intervention-wise? And Cindy Lewis as well down at Keys Marine Lab. And initially, I was somewhat skeptical, to be honest, that we could make a big impact and slow the disease down. But one of the things that we've tested with one of my graduate students, Aaron Schilling, really leading this project, is can we effectively use some of these intervention techniques to stop disease? And it turns out that one of them is way more successful than I would have ever anticipated. It's putting amoxicillin in this silicone base that was developed by CoreRx, a pharmaceutical company, and now a spinoff Ocean Alchemists. And when you take that base with the amoxicillin in it and apply it to the diseased lesions, it's over 95% effective in stopping that lesion from advancing. Now, other lesions can still pop up at the coral in other locations, so we still need to continue to do additional research on our intervention techniques and maybe come up with ways to systemically treat the corals or regions rather than just the individual lesion. But the fact that it was not just successful, but kind of wildly successful, 95% is a great hit rate for any kind of treatment. So the fact that it was that effective really kind of blew me away. And so now we and others are trying to think about, well, how can we scale this up in a way that's both effective and responsible? Broad scale antibiotic treatments are both difficult, and you have to worry about things associated with antibiotic resistance. So there's a great team of scientists that have all been working together on these questions and topics. And there seems to be strong support from both NOAA and the federal level and the state level as well. So there's been strong support for coral disease research that's been increasing over the past 3 or 4 years. And hopefully we'll continue to go on that trend. Here in the Keys, here in Florida, our reef resources are a really important contributor to our economy, to our recreation, to our fisheries, to our real estate market in terms of coastal protection. So anything that we can do to try to make sure that these reef resources are stabilized and persisting is really important.
So the last that I had heard about stony coral tissue loss disease was that we didn't know if it was a bacteria or a virus or what was actually causing the disease. This seems to me to indicate that it's more bacterial if an antibiotic is pretty effective at preventing its spread. Is that something that's been confirmed?
It has not been confirmed yet. So there are a couple different groups that are trying to work on identifying a pathogen. There is some evidence that it's bacterial. However, if you look at kind of the histological presentations, it's not kind of a typical bacterial surface-laden coral disease that we've seen before. It seems to maybe be coming up underneath through the coral skeleton into the living tissue that's on the veneer of the colony. And the fact that antibiotics can arrest a lesion, even though that suggests it's bacterial, it could also still be viral. You could just be killing the bacteria that were carrying viruses of some kind. But normally then you would expect like a rapid increase in lysis and viral spread in that region right afterwards, and we don't see that. So I think that there's pretty good evidence that it's bacterial. As someone who's studied coral diseases for a very long time, I also know that we could potentially spend years trying to figure out what individual microbe may be causing it. And it may be more than one microbe working together to create this pathogenic condition. Or it could be a number of different microbes that manifest lesions that have the same kind of disease signs, kind of all lumped together. So I think that work is important, and it may help us to target what kind of intervention we might do in the future, but we also don't necessarily need to wait to identify the pathogen before trying to take action now.
Yeah. Well, we're gonna get to those public questions now. I know I talked a little long with Josh. I just got very interested in all of his coral work. So we might stay a little long with those public questions, so keep sending them in. Our first one here is from Juliet. She says, how is diving on Bahamas reefs compared to Florida reefs?
Hey, Juliet, good to hear from you. So Juliet is one of our grad students at FAU Harbor Branch, who just recently successfully gave her proposal seminar. Well done. Bahamas water in general is much more clear than Florida water. And that's mostly related to the amount of terrestrial runoff that we get in Florida areas. The biggest difference is probably coral cover. So the areas of the Bahamas that have good coral development tend to have, 15, maybe upwards of 22% coral cover in some really great spots. The Florida Keys only has kind of 3 to 6% coral cover, and we think that that's almost been cut in half by the recent disease outbreak. So maybe down closer to like 1.5 to 3 or 4% coral cover remaining. The other thing that's quite different about the Bahamas is that there's more locations where you have really steep drop-offs near islands. So, one of the areas that we used to dive a lot in the Exumas was called Bach Wall. And it was a spot where, it dropped off from 60 feet at the crest and then just vertical wall face all the way down to about 900 feet. And you don't see much of that except all the way down past the dry tortugas in Florida. We have more kind of gentle sloping topography to most of our shorelines.
So in those drop-offs, then you get like, do you have corals growing on the sides of those drop-offs, or it's just nothing?
There's corals growing on the sides. Most of them tend to be smaller and encrusting just because you can't get, as they get too big, they'll topple off. But you'll also get plating. So since most corals have endosymbiotic algae and they're photosynthetic, they plate out to try to get more light as they get deeper and don't have as much access to light. So you see these terraces or shingles of corals that can be, 3, 4, 5 meters in diameter going all the way down those walls.
That sounds really cool. I want to see a video of someone diving down a wall, just watching that. I'm sure it's on.
Continue one.
Okay. Next question is from Amanda. What is one research technique that you wish you had more training in, or that you would recommend for students now?
That's a great question. So this is another Harbor Branch connection. Hi, Mandy. Mandy is a former student in my lab and was at FAU Honors College as well. She's now doing her Ph.D. at UCSD and was awarded an NSF fellowship to do that work, and she's doing great. I think there's probably a lot of things that I would recommend, or that I wish I knew better. But in terms of what I would recommend to students, there's two that I would say have changed remarkably during my professional career. And the biggest one of those has been a strong shift away from proprietary paid statistical analysis methods to open source platforms like R. So training yourself and there's all kinds of online training, editorials, tools, classes to improve your ability to use the R environment to do statistical analyses. That's probably the one that I would say is the skill or technique that I think is probably, for all kinds of students across multiple disciplines in science or social science, a really good tool to have. I would also say that we're tending to train more and more specialists rather than generalists. So, don't be afraid to do a couple of different internships or research experiences or volunteer in different labs or with different groups that are more varied and that are not kind of the same topic over and over and over again. Not only will you keep yourself fresh, you'll bring in new and fresh perspectives. So when I started graduate school, I wanted nothing to do with molecular science. But then I met a professor who was a forensics biologist, like trying to come up with a way to place or exonerate a person from being at a crime scene. And she was using this microbial profiling technique to profile the microbes on the soil at the crime scene and on the soil from a suspect's shoe. And kind of was this light bulb moment of, I can use that exact same thing in some of the coral disease research that we're working on. So if I had not been interacting with those kind of different groups and different ideas, I may not have picked up on that kind of new thing to bring into the field that I was in. So I think learning R and being diverse in who you interact with and what you do are probably the two things I would recommend the most strongly.
Definitely good advice. I tried to learn R. It didn't go so well. Actually,
You can still learn. You're still learning. That's what you mean.
yes. Some people that you know, Marilyn Brandt and Dan Holstein were my professors in grad
Yeah.
school trying to teach me R. And I was not their best student, I must say. But they were very good teachers.
Well, I also think that the amount of resources that are available relative to R is increasing exponentially. So like literally the online tutorials that are available now are nothing like what may have been available 6 months or 2 years ago. So even if you didn't get it the first time around, don't be dissuaded. Maybe the second time it'll take.
5, 6 years ago when I was trying to learn it.
Oh, it's very different.
Yeah, I
Very
probably should try and get back to that. Next question here is from Bailey. Could the amoxicillin research help revive the Great Barrier Reef?
Great question. Unfortunately, probably not. So the issues in the Great Barrier Reef that have really been problematic over the past few years have been bleaching. So when thermal stress, like too much heat comes in, that symbiotic relationship between the coral and the algae that live inside them starts to break down. And the algae get expelled out of the tissue, and so what's left behind is bare white coral skeleton. And if it's a short-term event, the coral can recover, and the few algae that are left in its tissues can grow back or can even recruit algae from the water column and recover. But if the thermal stress goes on for too long, then the coral will just die. And that's what's been going on in the Great Barrier Reef. So it's not a disease that can be treated with an antibiotic or some other kind of intervention treatment. It's stress related to basically global warming and local heat stress that have driven these successive bleaching events over and over again. So both dramatic measures to curb carbon emissions and slow the pace of climate change or reverse climate change, as well as research into kind of resilient corals and what species may be successful in a warming world are the two avenues that would be needed to help address what's going on in Great Barrier Reef.
Thank you. Definitely important to understand, and there's different stressors everywhere, but some of them we hopefully can fix.
Gotta have faith.
Yes.
In most cases, we know what to do. It's about developing the will and the means to do so.
Yes. Well, we're going to wrap things up now. Thank you, everyone, for joining us. And thank you, Josh, so much for being a part of us. I know you're on vacation, so thanks for joining us anyway.
Thanks for having me, Madeline. And I thank you all for taking the time to stop by today.
Well, we are going to have one more Behind the Science episode before we go on a bit of a hiatus. So definitely tune in next Wednesday, June 24th at 4 p.m. Our guest is going to be Dr. Daria Akainak. I've been practicing that. I'm sorry if I got it wrong. She is a mechanical engineer and an oceanographer, and she's doing a lot of really cool work trying to solve problems with underwater imaging and with computer vision, which I'm not sure quite what that is. But she is trying to work on specifically making underwater images and photographs better in terms of lost color and contrast that happens because of the way that light moves through the water. And she also, along with that research, is working to try and understand how non-human animals see the underwater world. And so she uses a lot of different cool techniques to try and get a better understanding through those other animals' eyes as well. So she's going to be a really cool guest. Tune in, like I said, next Wednesday, June 20th at 4 to hear more about her. And then if you want to see some of our other Behind the Science episodes that have happened previously, or if you want to check out some virtual tours of Harbor Branch that we have coming up, definitely check out our website. You can find a lot of really cool research and really wonderful opportunities to learn more about them. So hopefully we'll see you guys again next week. and you have a wonderful Wednesday evening. Bye.